Not much modeling, but really it's like a discovery-based research. The title of my talk is about non-coding arrays. So I don't know how much you know about it. I'm going to walk you through. So this is my outline. So I'll give you a little bit of background about what we do in general. And then I'll give you the kind of a short story about what we found. And then last, if I have time, I want to kind of introduce our lab because we're looking for a good computational person. So it's just one of the reasons. So sometimes I run over time, so I'm going to kind of acknowledge the people involved in this project first in case I don't get time. So this is a big lab that I'm in, over 40 people. And these are the people that played a significant role in this study that I'm going to describe. And all of the experiments were done at UNC. So they do this mouse breeding and infection, and we get the samples, we do the analysis. And also we have a very good industry collaborator with Illumina and former Merck Rochester. So we got lots of industry support. So our lab is using system biology approach to study infectious disease. And this is our life cycle. So basically we started with many model systems and we collect lots of data just as Professor Wu just described with both the phenotypic data and the molecular level high super profiling. And then we process the data and put them together. We want to model this data. And ideally we want to narrow down the big hairball into something that we can grab on it, and then we can design a new layer of experiments, and then keep going to finally we can find a drug target or some mechanism. And at some point we say, well, maybe you're good enough, we want to go to the clinical. So that's our kind of general workflow. So one thing that I'm going to talk about today is that we really are interested in host response. I mean, virus passage is very important, but we're really interested in the host side of the story. Particularly, today I'm going to talk about the transcriptional regulation of host response. The reason is that because the technology, the microarrays, we have learned a lot from the transcriptional regulation of the host response, and we find it's very important. But still, it's not enough because we want to go from profiling, from description, what is happening to the mechanism of the disease. What is the mechanism behind the viral pathogenesis? Really, in the lab, our PI is really interested in this particular question: how virus kill the host. That's what he's most interested in, how it kills the host. I mean, of course, there's a kind of a really social kind of economic impact, especially if you look at AIDS. Just still, after how many years, we still don't have a vaccine. We spend so much money. It's just kind of frustrating for everybody. So typically when we do host response, we do transcription regulation. Usually we run microarrays. People know about arrays that usually you spot your probes on your array, and the probes were designed based on what you know about the genome. So what you know about it, you put on the array. If you don't, forget about it. But really, if you look at the genome, so one of the notions is people say the genome is pervasively transcribed. And people say somehow probably just a transcription noise, but if you look at it, 5% of that genome that are really conserved, and there's a really good reason to believe those things are functional, but for this 5%, only 1.5% of the genome is a protein coding genes. Still, majority of them are non-coding. People don't study them, it's just dark matter. So, do you care? Probably you should. So I'm going to give you the reason that why probably you should care. So when we started, so there's a kind of a technical background is that we do RNA-6, that was like 2009. So basically, the idea you take your total RNA, you fragment it, sequence them, you don't care what is there. And then later you map to the genome, And then if you have your annotation, you're gonna just count them and use this as a strategy to quantify the transcripts. Some cases you may say, "Oh, I don't care. I'm not happy with the genes you annotated. I'm gonna do my own transcript assembly and then put it into this kind of same process. You do your quantification." So typically people call it next generation sequencing or deep sequencing. Really, we're doing RNA sequencing, not a DNA sequencing. So we're using this approach to do the transcriptome profiling. But really the nice thing about it here is that what this implies that you can profile the genes, the transcript you don't know before, but your data has that information. Really what you need to mine that data to find out what's in your data. So for example, what is nice about it is, for example, I have this gene structure here. This is the information that I get. This is the SARS-infected samples. And this, you see the correspondence. It's like, not only I get a quantification of the whole transcript, I can get a structure of the transcripts. If you look at the isoform switches and the splicing, you can get this information because it correlates really nicely with your transcript structure. So you have a much finer resolution about your transcriptome changes. So the system we're using is, I guess, I'm not sure everybody knows about this project. It's called a Collaborative Cross mouse project. Basically, it started with 8 founder mouse strains. They have very different genetic background. What you do is just keep crossing them until you get many kind of pure lines. They're very, in terms of their genetic backgrounds, are very homogeneous. And the idea is that at least the pool of this line will capture 90% of the genetic variation of the mouse population. The good thing about this is really good model to study human population because none of the 2 people are the same in terms of their genetics. So if I put say a 1918 virus in this room, probably some will die, some will be really happy. So there is a genetic determinants behind this disease outcome. So this is a good model to study the infectious disease. So we're working on this. So as a kind of a starting point, we just only take a 4 mouse strains. We infect with the SARS, we collect the typical data as we do, and then we do our profiling. In this case, we don't do the array because the mouse strains are very different. There's no way that your single array platform will work on this study. That's why we go from sequencing. And when we got it, it's a headache. Well, how am I going to analyze these things? And then we have to kind of go back to find develop a computational strategy to make sense of this data set. But the nice thing is when you look at this data, it's really cool is that you can look different classes of transcription. Really, I'm going to talk about today is, if you look at here, there are tons of data goes to intergenic. Basically, this is the transcription activity is between typical person profile, say it's a coding genes. This is beyond your coding genes. It's a pretty significant percentage. So this could represent either a novel coding gene or some non-coding genes, but really what they are. So first we say we just go through literature, go through database, looking for the annotation, what they are, and then we found about 10,000 those annotated, maybe partial annotated, non-coding RNAs. We found about 500 show difference. I don't care about the coding genes in this particular case. And then also, because the nice thing about a sequencing, you sequence them, doesn't matter you know or not, and then you can later do your analysis, you find those unannotated regions. You find over 1,000 unannotated genomic regions. So basically, we have no idea what they are, but they show difference after SARS infection. So this just tells you that there are more things in the genome that you probably should be aware. And one of the questions is this specific to SARS infection? So this is the data that we just take 37 random choosing things, and then we run a PCR on the replicate samples. And not only we do SARS, we do flu, the same strains. What you see is we got a very good correspondence between from our original sequence and the replicate samples. And I think it's yours to see in the influenza. So this is not just about SARS. The second thing that in the lab, we're really interested in innate immunity. Particularly in this study, we take out these two kind of important players of innate immunity, see what is going to happen. So basically, we take the mouse, the wild-type mouse, we knock out this one or interferon, and infect with the SARS to see what happens. What you see you have very different kind of a cluster of the non-coding RNAs. Some show consistent changes, some up or disappear. Like you see here, are really strong and then it disappears. So basically, there is strong evidence that this non-coding RNA is not like some noise. It has a very strong relationship with innate immunity. Further, because people criticize and say, well, you profile whole lung, you know, there's so many immune cells infiltrating the whole lung. How do you know what is what? So what we do is we take the embryo fibroblasts from the same mouse strains. We either infect with influenza or treat it with interferon, and then we just profile them again. What you see that they respond to interferon treatment in vitro. They also respond into influenza in vitro. So really, this just tells you there's a really strong. Yes, go ahead.
I'm sorry to interrupt, but did you say how long these RNAs are?
How long? Non-coding RNAs?
Yeah, well, you call them long.
Yeah. So typical, when people say long, it's over 200 base pair.
100 base pair?
200.
Oh, 200, yeah.
Yeah, so that's the kind of convention in the field. If it's shorter than 200, people call it shorter ones. Yeah. So we publish this, and then we continue looking for other things. So for example, this is another study we do HIV, infect the CD4 T cells. You see this is another human non-coding RNAs. So this basically this kind of pic shows the strong, the strength of the expression. So you see this infected have a very strong upregulation in the mock. They're not much. So my prediction is that many viruses can induce these non-coding RNA expressions. We just haven't looked at it. So what do we learn? Long non-coding RNAs are involved in viral host interactions, and many of them probably are to be found. What about those small ones? Because you just asked me those long ones, what are the small ones? So the small ones are usually below 200 nucleotide. There's no kind of a very strict definition. It's just because I think it's because of collagen RNA isolation, the column, whatever. Some reasons that people just stay with that. Basically, the small ones, the most well-known ones is the microarrays, 22 nucleotide. And there are others like small nuclear RNAs. People probably don't care that much. So in this case, we go back to these full strains. In this case, we infect SARS and the flu. Do our normal practice, collect the data, analyze them. So short story is we publish this because we found over 200 small non-coding RNAs. But the interesting is they're from different class. When you look at the small RNAs, typically if you go to literature, people say, I'm looking at microarrays because it's well known, their mechanism has been well studied. But what we found is there are many non-microarray small RNAs. Because people know a lot about the microarrays, so I'm going to do some literature. This is a list of microarrays that we found show difference after both virus infection. And I found this is after we published it. I just doing something else. I found this review. I said, well, this people looks like people know a lot about the microarrays, about the immune system. When I checked the list, pretty much we cover most of them that people know about it from a single study. Beyond known microarrays, we also found unannotated. So for example, there's a small RNAs in these introns. Same here. It shows difference in different strains, but we don't know what they do. The bad thing is we don't know what they do. But the reality is there are more things to be discovered. So the bottom line, I think host response is more than those 20K coding genes that people typically study. And my suggestion is, if you have array data, probably it's too late to change it. But you just beware if something that you cannot explain, there's another layer of the variable that might be involved. If you do have RNA-seq data, maybe it's a good idea to reanalyze it. So I'm gonna, so any questions so far? I'm gonna, otherwise I'm gonna just, yeah.
Yeah, reanalyzing the data. I've started to look at some of our RNA-seq data with this popular program called Couplings to try to find the non-annotated stuff that's changed after vaccination. And mostly what I'm finding, it's very difficult to be convinced that a lot of this stuff is novel. They seem to be very minor variations of start and stop sites of annotated RNAs or little splicing variations, tiny ones, and the software keeps calling it a novel gene, and it doesn't name it with the same gene that it should be naming it. So do you have suggestions for the best way to reanalyze it? Because I'm not very happy with what's out there.
Yeah.
Without writing our own code.
Well, that's what we do. We write our own code because, I mean, and at the beginning I said we're hiring people because we got tons of data, and we know that we are not exhausting the value of the data. I mean, the tools you talk about, it's a very popular tool, but it's not the final thing that you still have to come up with your own kind of a customized solution to address. Because, I mean, the point you raise is that your data is telling you that the existing annotation is not correct. So how do you correct them? That, I mean, at this point, the current status is a fast evolving field. People are trying to do better, and there are computational needs. There's also technical needs that how to analyze, how to sequence your sample based on your needs. Are you going to improve your annotation? You're going to do a better quantification. There are different considerations. So I don't have a very simple one word, like a one sentence solution, but definitely something that if you think there's a strong reason that there are things that you think they're valuable, I think you should spend more time on that to find out the good people to do that. Yeah.
So you mentioned briefly the microRNAs.
Yeah.
And so I was wondering if you've looked at all at the coding genes,
yeah,
coding genes, and which microRNAs might correlate highly with them, especially possibly different splicing variants. And then you could identify and also use like motif recognition to see if you could find like novel modifiers, novel microRNAs that control gene splicing.
So I don't have the slides here. We do to the microarray, mRNA regulatory network modeling things. So we have a separate study, and also I didn't put it here. So basically, if people know about the microarray, basically many of the microarrays are from the introns of the coding genes or the introns of the non-coding genes. And the microarray biogenesis, one of the first steps is the transcriptional regulation of the precursors. So there are many steps to regulating microarray biogenesis, and the microarray will regulate the transcripts and then also protein production. So there's another line of a big regulation that is the study that we're working on.
So all those non-coding RNA that are published?
Some of them are, but I mean, I don't know what you mean published.
So ones that, the novel ones that you found,
Yeah.
are they published?
Oh, you mean from our paper?
Yeah, from
Yeah, it's in the
FAST
supplementary
format?
table.
Okay.
Yeah.
So in FASTQ format?
Yeah, we have the genomic coordinates that you can go there, just extract out the sequence that you want. Yeah. Or you can load the browser or whatever. Yeah, we're trying to deposit all of our sequence data into NCBI before we analyze it because it's just too much information. I think it's better for everybody else to mine the data together. So, yeah. How stable they are? You mean? I guess from the heat map I showed, they have a different time kind of profile. So I mean, probably everyone has its own kind of a profile. Yeah.
Other questions? One more question.
Yeah. What proportion of, I know you showed interferon, gamma, and STAT1.
Yeah.
As functional significance based on your non-coding RNAs. What proportion of your non-coding RNAs have you so far found any kind of significance with respect to coding gene? Like, I think you showed 2, right? And STAT1 and interferon gamma.
So we knock out a STAT1.
So the knockout one.
So basically, we take a mice, knock out a STAT1, and then do the infection. And then another is we take out an interferon alpha receptor and do the infection, and then profile them before and after.
Yeah. So my question, I got that. But my question is, what proportion of your, I don't remember the number of ncRNAs that you identified.
Oh.
What proportion have you shown? Because what could it be just that the virus just hijacks RNA polymerase and starts making all these ncRNAs?
But I don't have an estimation about this particular, because for that, the data I showed on the knockout, we only did a PCR about the 37. Right. We didn't profile them. If we profile the knockout ones, then we will know. So we have other studies. For example, for this CC, now, I mean, what I described, we only sequenced 4. Now we're sequencing over 8, over 120 samples. So now we have a much bigger study because the technology is more mature, the methods are more mature. And also, I'm going to describe, we have a more infrastructure to support this kind of analysis. So probably maybe in a year or 2, I know the percentage.
No, I was just curious, could it be just not a real cause and effect thing, but just happens to be that the virus is hijacking the host RNA polymerase and transcribes more of the ncRNA?
It's possible, but I don't think it's all that, because people study non-coding RNAs. The non-coding RNA is just like a typical gene. It has its promoters, get a regulated transcription factor. And for the HIV study, we're doing the transcription factor study of those things. So it's just like a typical gene. And then the reason is that I don't know why people just don't study them. Probably it's hard.
So it's not that people haven't studied them, it's just that now we have the techniques that you can do 230 million reads that you can actually find them. Right?
Yes,
Yeah.
that's, yes. Yeah. Okay, so I'm going to do a little bit of self-promotion. So this is our lab, and we have many people, and the people coming from very different background, from biology to computer science, and we work on many viruses, and we have our kind of a standard Microarray workflow set up in the lab. It's all automated, and we are doing our own sequencing in the lab, and we have our own proteomics, the mass spec machines. And we set up our own server room to store the data, manage the backup, and processing this thing. And our computing group have about 10 people now. We have a faculty. This is myself, and then we have 5 postdocs with a different background, and we have a PhD student in computer science. Have staffs that work on different aspects, and also I have to open this now to the computation. And the goal is we're trying to shake up the field using the system biology approach. Thanks.